Okay, ladies and gentlemen, for today's podcast, we want to actually talk about this structure of DNA. We've spent a lot of time talking about the alleles, and specifically, like, what were the alleles, what's the probability of them getting passed on, how do you get alleles from your parents, how do they translate into traits. And then we talked about how the alleles are actually sections of chromosomes, and the chromosomes are coiled up in DNA, which you find in the nucleus of your cell. So this, of course, begs the question, well, what is DNA? Well, the first thing to talk about is what the Na stands for, which is nucleic acid. So DNA is a nucleic acid, which, if you go back all the way to like September and October when we were talking about it, it's one of the organic macromolecules. So if it's an organic macromolecule, that means that it has a monomer unit and a polymer unit. So the polymer unit, the pieces that are joined together to make the overall molecule, are actually known as nucleic acids. There's two types of nucleic acids. There is deoxyribose nucleic acid, which is what we know of as DNA. Okay. There is also ribose nucleic acid, which is another type of nucleic acid called RNA. You may have heard of RNA. As we get into some of the later stages of this process, we're going to discuss RNA in some detail. So that's the polymer unit. But one of the things that we picked up when we were discussing all of our organic macromolecules is that all of our polymers are made of monomers. And the monomers for both DNA and RNA are known as nucleotides. So if I assemble a bunch of nucleotides together in a chain, I'll end up with either DNA or RNA, depending on how they're assembled, which nucleotides, and what structure they take. So let's take a little bit of a closer look at a nucleotide. So this diagram right here is a nucleotide. So right here you'll notice that we have a 5-carbon sugar. You see these numbers 1, 2, 3, 4, and 5. Okay. We have a phosphate group. And then over here we have what's called a nitrogenous base. So basically, all nucleotides are made of a sugar, a phosphate, and a base. What makes one nucleotide different from the other is the base. So all the variety in nucleotides comes from our bases. Okay? And you notice here that there's a number of different types of bases, and we're going to talk about that in a second. So let's actually do that. So we've already hit the nucleotide and the nucleic acid are the polymers versus the monomers. So what we have here in this basic nucleotide are the three main components, the phosphate group, the sugar, and the base. For the bases, there are actually 4 types in DNA. Okay, and they are known as the pyrimidines and the purines. Okay, so the nitrogenous bases, or the nucleotides based on the nitrogenous base, are divided into 2 main groups, the pyrimidines and the purines. The purines are the adenine and the guanine, and the pyrimidines are the thymine and the cytosine. I'm sure you've seen these before. You're probably familiar with the terms adenine, thymine, cytosine, and guanine. When we write these out, we frequently refer to adenine as A. We frequently refer to guanine as G. We refer to thymine as T, and to cytosine as C. So that's where you get this, the nitrogenous base, ATCG. So for each of these nucleotides, the sugar and the phosphate are always the same. What makes one nucleotide different than the other is this. So this is where the variety comes from. So if you want to have an adenine, the sugar and the phosphate group are the same as every other nucleotide, but the nitrogenous base is slightly different. Okay? So one of the things and the reasons we watched the Race for the Double Helix movie is to talk about how the nucleotides connect. Scientists knew for the longest time that they were in there, that they came together, that they formed a organic macromolecule, but what they weren't really familiar with is the structure they took when they connected. And it was really interesting because, once we went through a lot of Erwin Chargaff's work and we learned about the 4 bases, there was a lot of question, like how do they come together? And at the time, our technology was sort of limited to X-ray crystallography, so we couldn't really see it. We had to sort of divine what it looked like based upon those crystallography images. And that's why Rosalind Franklin's work was so important and Maurice Wilkins' work was so important. And it actually was a whole reason as to why Watson and Crick's deduction as to the actual structure was so brilliant. So what it turns out is that the nucleotides, hold on one second. What it turns out is that the nucleotides connect into a shape that's very common in nature called a helix. A helix is a lot like a slinky. So if you were to take a slinky and stretch it really far, it would look like this. And that's actually what a helix is. A lot of our protein structures actually have a helical shape. It's very stable. It's a common structure in nature. What makes DNA so interesting is it's what we call a double helix, which means it actually has 2 of these helices sort of running side by side. So there's one, and then DNA would actually have another one that runs side by side. So that was one of the first things that Watson and Crick came up with that was so genius. The other thing they realized is, well, where do all the pieces of these nucleotides fit? So first you have the sugars and the phosphates. So what do they do? And what it turns out is that it is the sugars and the phosphates that connect the helix chain. In fact, we refer to this as the backbone. So what happens is, if I have this as a helix, the line that I've drawn is actually the sugars and the phosphates connected to each other. And the nitrogen base sort of pops off of it like that. And that was a big deal because the other thing that Watson and Crick realized is that the bases face each other. So if this is my double helix, I were to look at one chain, there's all the sugar phosphates, and then here's my other one, and I have all my sugars and phosphates. And what would happen is the bases of this chain and the bases of this chain would connect to each other. And that's actually what holds the 2 chains together. The nitrogen bases connect with what we call hydrogen bonds. Hydrogen bonds are a type of intermolecular force. They are very weak individually, but as a unit, but when you actually look at them in groups, they're very strong. And that actually is what keeps the DNA together. And you can actually look at this over here. All right, so if I look at this diagram, this line right here, that's my backbone, my sugars and my phosphates. Okay, and then when I expand the image, you can see here I have my nitrogen bases holding each other together in the middle with hydrogen bonds, and out here I have my sugar and phosphate backbone. And the natural shape this takes to make the connection work is that helix. Now, one last thing I want to talk about in this particular presentation is that you'll notice that the two nitrogen bases that are connected are not the same. It turns out that the bases are complementary to each other. So what that means is that the bases do not actually connect with a similar base. So adenine does not connect to adenine. Thymine does not connect to thymine. What instead happens is adenine always connects to a thymine. Adenine and thymine are complementary. Cytosine always connects to guanine. Cytosine and guanine are complementary. And so one side of this helix is complementary to the other. One last thing is that each side runs in opposite directions. So one side of the helix runs up, the other side runs down. Okay, so those are some of the basics of DNA structure. Hope you enjoyed it.